Hi, I'm Elizabeth Williams, conference producer at Technology Networks, and I'm here to introduce our next talk, Unlocking the Epiproteome, Next Generation Biomarkers for Immune-Mediated Diseases. And I'm very pleased to welcome Dr. Aaron Javitt. Aaron is the Director of Computational Biology at Promise Bio, where he partners with pharmaceutical companies to uncover clinical insights from epiproteomics. He studied at Princeton University and earned a PhD in systems immunology from the Weizmann Institute, where his work revealed how post-translational modifications shape disease, with publications in journals such as Nature Biotechnology and Nature Cancer. A very warm welcome to you, Aaron. Following the presentation, we will also have a Q&A session, and we welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. I will now hand over to Aaron to begin his presentation.
Thank you so much for that really kind introduction. And of course, thank you to the organizers for the invitation to speak. As was said, my name is Aaron Javitt. I'm the Director of Computational Biology at Promise Bio. And today I'll be discussing how we're trying to unlock the epiproteome by pushing the boundaries for biomarker discovery for immune-mediated diseases. So just a quick word on our company. We were founded a little over 2 years ago. The founding team is Dr. Asaf Kassan and Dr. Anil Vexler, along with our scientific co-founder, Professor Yufat Merbel. We're a multidisciplinary team of biologists, clinicians with over 30 years combined of proteomics research experience. And we have both a list of strong strategic investors and clinical partners. Our main goal as a company is to poise post-translational modifications, or the epiproteome in general, as the next forefront for precision medicine and biomarker discovery. So to start first, I want to take you through a quick all the way back to intrabiology and the flow of information in the cell or in biology. Traditionally, we think of the information as going from the genome or the genes in the cell through transcription to the transcriptome, following translation to the proteome. And most of the current solutions whereby we have to measure what's going on in the cell stop at the level of protein abundance. However, our biological processes do not stop there. Following the translation of the proteome, many proteins are modified, and these post-translational modifications are critical for regulating protein activity, location, and timing of functional processes. And it's this level of the epiproteome, or the collection of all of our proteins and their post-translational modifications, that ultimately dictates the functions of our cells, our tissues, and disease phenotypes. Just to give you a sense of a few of the PTMs and how important they've been as clinical biomarkers, I want to go back to 1970. So in 1970, it was discovered that glycated hemoglobin, or a glucose fused to hemoglobin in the blood, serves as a more stable measure for glycemic control. And this is one of the first examples where a post-translational modification on a protein was identified as a better biomarker for tracking disease severity. Going forward around 2 decades, we get to anti-citrulline protein antibodies in rheumatoid arthritis. So during the course of the development of rheumatoid arthritis, our bodies start to make hypercitrulated proteins, or they accumulate many citrulline modifications on the proteins that circulate in our body. And one of the critical diagnostic tests for rheumatoid arthritis is detection of autoantibodies, antibodies against ourselves, and specifically against citrulinated proteins in our blood. And finally, jumping forward another 2 decades to 2020, we have phosphorylated tau in Alzheimer's disease. Phosphorylations accumulate on tau in the brain, and this ultimately causes the formation of these neurofibrillary tangles or plaques that form in the brain. So phosphorylated tau is both a mechanistic driver and an early detection marker. While all three of these clinical biomarkers are PTM-driven, they occur across different diseases. But they have one thing in common. It took years of painstaking research to discover and identify them. What if there was a way whereby, with a fast, broad way to profile PTMs, we could uncover and identify PTM-driven biomarkers across a variety of diseases? Enter Promise OS Suite, where we pose the first cloud-based platform for unbiased, broad epiproteomic analysis to enhance drug development. So for the first part of my talk, I want to discuss a little bit about how Promise OS works, and then I'll move into a variety of use cases across immune-mediated disorders to show how augmenting our data with PTMs can better provide us biomarkers, mechanism of action analytics, etc. So the Promise OS suite is divided into 3 stages. The first stage, or the capture stage, uses a pipeline we call Prospect for broad epiproteomic profiling. We can identify over 60 types of post-translational modifications from standard mass spectrometry data, thereby maximizing the yield from every single experiment. Following this, we have the augment phase, where we leverage Epitome, the largest data link linking PTMs, functional states, and clinical outcomes. And finally, on top of this, we apply our reasoning models, whereby we can derive actionable biological insights to empower decision-making. So first, a few words on Prospect, which is our platform where we identify post-translational modifications from mass spectrometry data. It's important to note that we do all of this without chemical enrichment. Traditionally, when one wants to identify PTMs in proteomics, they go through the painstaking process of extracting and amplifying a certain type of post-translational modification, such as phosphorylations or glycosylations. Here, we say, let's understand the landscape of PTMs in an unbiased manner. We'll take the full proteome, all of the proteins, and all of the PTMs in a sample, and we'll go and identify over 60 types of PTMs on their endogenous levels in the sample. This method was published by myself and Dr. Kassen in 2023 in Nature Biotechnology, and was developed in the Merble lab at the Weizmann Institute of Science. In brief, the way this method works is as follows. We start with a sample processed through traditional mass spectrometry techniques, often digestion of the peptides from proteins, and traditional LC-MS/MS acquisition. And in a conventional analysis, what would happen is these peptides would be matched against the canonical proteome, or all of our protein sequences, without post-translational modifications. And as you can see in this little schematic, there are many peptides that existed in our original sample that aren't identified because they don't have a reference in the proteome. From this conventional analysis, one can get protein abundances in our sample. However, what we do in Prospect is, rather than matching just to the conventional proteome, we add in over 60 types of PTMs, and through a combination of iterative searches, are able to identify many of the post-translationally modified peptides in our sample, thereby getting results that incorporate not just protein abundances, but also modification states, or PTMs. We validated this method extensively in our publication in Nature Biotechnology, and we did so in 3 discrete ways. The first was through experimental confirmation. We showed identification accuracy of our method against 3,000 synthetic modified peptides. We know what peptide is in the system, and we were able to show that our method can accurately identify them. We also did the reverse. We took 20 novel cancer-specific modified peptides that we identified and experimentally validated them by synthesizing them and injecting them into the mass spec and confirming that the spectra were the same. The second way is through performance benchmarks in comparison to competitor tools. We found overall we were on average 54% more accurate, on average 25% higher resolution, 25% more identifications, and 11 times faster runtime than our competitors. And last but not least, we took our findings and compared them to published evidence. So we compared them to public post-translational modification databases. We cross-referenced against documented mutational variants, and we were able to do literature confirmation of post-translational modification motifs. All told, we were able to show both experimentally and through comparison to the literature that what the PTMs we are identifying are truly in our sample. So I want to give you one sense of the scope of how much data is being lost in traditional mass spectrometry search and how many insights we can add. Here in this is one specific cohort of urine proteomics. And here we're showing the average number of peptides detected per sample. Here we see using conventional off-the-shelf mass spectrometry search, we identify around 1,000 peptides per sample. And this is with base parameters with no post-translational modifications included. When we add in over 60 PTM types and do a combination of iterative searching, whereby we fit the PTMs we identify to the specific experimental and disease setup, we're able in the end to get over 5,000 peptides per sample, thereby increasing by 5-fold the number of peptides we can identify per sample. In other words, if one were to take these data and analyze them with conventional mass spectrometry, 80% of the information the machine captured would be lost. And only by incorporating PTMs can we truly leverage all of the data we've captured. These PTMs that we've added in this specific cohort represent 23 different groups of post-translational modifications across our proteome, across many protein sites, and different amino acids. Now that we have all of this great data, the question is, what can we do with it? On the one hand, more data is great. On the other hand, more data can often be confusing. We believe that these data help us to better understand the specific tissue we're looking at. So if we think about a given sample, we oftentimes refer to the proteins present in a sample. And our assumption is each gene encodes a protein. But over the past 10 to 20 years, this has been found to not be the case. Most of the proteins in our body exist in many different proteoforms, or the same protein with different combinations of post-translational modifications sitting upon it. In conventional mass spectrometry analyses, the peptides lose their connection to the original proteoform. We use trypsin or other methods of digestion. We know which peptide is assigned to which protein, but not the intact proteoforms as they existed in the body. Traditionally, all peptides with a specific sequence would be assigned to the parent protein, which is a kind of Frankenstein composite of all of the proteoforms that existed in that sample. And so traditionally, one would get protein abundance values. For every protein in every sample, they would have an average measure of all of the proteoforms that existed in that specific sample. We immediately can add on to this protein layer the layer of post-translational modifications, PTMs. And in this case, in this hypothetical example, we have 5 distinct sites of PTMs across the protein. For each of those sites, we can say what PTM is there, which position it's at on the protein, what amino acid it's modifying, and how much of it there is relatively in the samples across our cohort. And this will be the second layer of information we can extract from PromisOS. The third layer is computationally reassembling proteoforms as they existed in the sample. The way we do this is we cluster all of our peptides into discrete signals from a cohort. In this case, one can imagine getting 3 distinct groups of peptides, which we'll label form 1, form 2, and form 3. For each of those forms, which have a cohesive signal, we can understand which PTM sat on that form and how much of that specific proteoform existed in the sample. These 3 layers of information, the protein layer, the PTM layer, and the proteoform layer, comprise what we refer to as the Epiproteome, or the collection of all post-translational changes that occur to the proteome from the time it's been translated onwards. And as I said initially, these layers of information bring us one step closer to understanding the function of our cells our diseases, our tissues. Now that we have a sense of the types of data we can extract using PromisOS, I want to walk through a number of different use cases. These use cases will highlight many of the ways that epiproteomics enhances our understanding or biomarker discovery across immune-mediated diseases. As is the case with all of these use cases, these are all reanalyses of publicly available data, which means these samples were collected without PTMs in mind, with no special enrichment. All of the insights that we're showing are by computational reanalysis of these samples. The first use case I'm going to discuss is identification of citrulline signatures in rheumatoid arthritis, whereby I hope to show that epiproteomics provides novel markers to define disease states. For this, I'll be showing the reanalysis of two cohorts. One is serum samples published by Jahangir et al., and the second is reanalysis of synovial fluid from Mahendran et al. First, starting with our serum samples, we extracted all of the PTMs from the mass spectrometry data, and using unsupervised clustering, we could see that the samples divide into two distinct groups. The group in gray here are patients who are, or individuals who are healthy. These are serum from healthy individuals. The group in red are patients who were clinically defined as having rheumatoid arthritis through double positive status of rheumatoid factor and ACPA, or these anti-citrulline protein antibodies. While it's no great feat to be able to separate serum signatures from RA in healthy patients, as this is a very systemic, very inflammatory disease, the better question in our mind was what is driving the separation, and more specifically, which PTMs are driving the separation. As I mentioned at the top, we know that citrullination is a key disease driver in rheumatoid arthritis, and anti-citrulline protein antibodies are one of the key biomarkers to date. And we wanted to know whether our broad, unbiased PTM profiling would recapitulate these same findings. So what we did is we asked, across all of our different types of PTMs, which slice of data, which PTM type, is best separating RA individuals from healthy individuals. And we've ranked all of our PTM types here based on the distance that they cause between the RA patients and the healthy individuals. As you can see, citrulline is ranked the top, meaning that the citrulline PTMs are the greatest separator of RA patients from healthy individuals. Here you can see a PCA or dimensional reduction of the same patients using only the citrulline sites alone. We're taking only the citrulline PTMs, and these sites alone are sufficient to separate RA patients from healthy individuals. If we were to take other PTM slices on the list, such as oxidation or O-glycosylation, which are ranked more lowly, you can see that there's little to no separation between the RA patients and the healthy individuals, suggesting that citrulline is unique in its ability to distinguish disease status. Going one step further, we were able to use machine learning to derive a score of all of these citrulline sites, distilling down to the sites that best separate double positive patients from healthy individuals. Markedly, while this score was trained on double positive or RF positive, ACPA positive versus healthy individuals, when we apply this score to the other samples in the cohort, namely single positive ACPA or single positive RF, we see a gradient of patients from double positive all the way to healthy, suggesting that this score not only fits the data it was trained on, but can be extended to define a gradient of rheumatoid arthritis status from patients who are double positive all the way to patients who are double negative, and thereby serve as a potential future marker for defining RA subtypes. Taking this even one step further, we wanted to see if our findings from serum or circulating proteins could be extended to sites closer to the organ impacted in the disease. And to do this, we reanalyzed a second data set of synovial fluid from the joints. And in this data set, we had two diseases. We had rheumatoid arthritis here in red and psoriatic arthritis here in blue. Now, distinguishing psoriatic and rheumatoid arthritis is a really big challenge. On both the protein level and the PTM level, unsupervised, they don't separate. And this isn't surprising. RA and psoriatic arthritis are both inflammatory-driven arthritis with highly similar PTM and protein profiles. Nevertheless, through supervised clustering, we were able to define a subset of our PTM sites which do differentiate between RA and psoriatic arthritis. Beyond that, when we ask again which PTM types are most driving the separation, we see citrullination emerging again as the top-ranked PTM site. And deriving again a machine learning score, in this case from synovial fluid, we can use these citrulline sites to significantly distinguish between patients with RA and patients with psoriatic arthritis. Perhaps most interestingly, when we compare these two markers, these two citrulline scores, one from synovial fluid and one from serum, we identify a common feature to both of them, which is citrullinated complement C3, which emerges as a novel marker of rheumatoid arthritis. In the case of synovial fluid, it aids in distinguishing RA from the very similar psoriatic arthritis. In the case of serum, it defines different subtypes of RA from double positive to double negative, and serves as a marker that's both circulating and identified at the site of the local disease. This shows that epiproteomic profiling or broad PTM profiling can first of all recapitulate much of what we know about RA and citrulline over painstaking years of research with simple reanalysis of data. Beyond that, we're able to identify PTM-driven biomarkers that seem to poise us to better use citrulline sites to differentiate RA from psoriatic arthritis and different subtypes of RA. Moving on to the next use case, I want to jump tissues. We're now going to be discussing colon biopsies in a different disease setting, ulcerative colitis, to show that here epiproteomics highlights core disease-driving mechanisms. So rather than focusing here on biomarkers, I want to focus more on mechanistic insights into ulcerative colitis, and specifically to look at JAK-STAT modulation. We reanalyzed data from Yanker et al. From 16 biopsies of inflamed colon and 24 biopsies of non-inflamed colon. Through our reanalysis, we identified 5,800 proteins and were able to augment that with over 16,000 post-translational modification sites. These PTM sites, as similar to our citrulline cohorts, span many different types of PTMs, from methylations and acetylations, all the way to phosphorylations, ubiquitinations, and simulations. We can use this breadth of data to better understand how proteins change in inflamed state in colon. And I want to take a really canonical example of a protein known functionally to drive inflammatory processes in ulcerative colitis. And that's the example of JAK1. I want to focus here on JAK1. JAK1 is a highly targeted protein in autoimmune diseases and specifically in ulcerative colitis because it's a master regulator of cytokine production and inflammatory response through the JAK-STAT cascade. Nonetheless, while we know that it's functionally important, if one examines JAK1 at the level of protein abundance, we don't observe any change between inflamed and non-inflamed colon. Here in this volcano plot, proteins that are increased in inflammation are found in this quadrant. Proteins that decrease inflammation significantly are found in this quadrant. JAK1 on the level of protein abundance sits right in the middle. Were it not for the years of research categorizing JAK1's function, we would not know from these data that JAK1 is involved in inflammatory responses. However, if we take our post-translational modification sites and perform our proteoform clustering algorithm on this, we can identify two distinct forms of JAK1. In these diagrams here, we can see all of the PTM sites on JAK1 form 1 or JAK1 form 2 from the beginning of the protein to the end. JAK1 form 1 has many modifications across the protein. JAK1 form 2 has fewer modifications, although notably it has 2 phosphorylation sites towards the middle of the protein. When we then look at changes in proteoforms rather than full protein abundance, we can see that JAK1 form 1 is significantly decreased in inflammation in colon, suggesting that if we add on the layer of PTMs and proteoforms, we can understand or identify changes that we know to functionally occur in these tissues. Going one step beyond this, we can do this not just for JAK1, but for all of the proteins involved in JAK-STAT signaling through analysis of one of the pathways of cell surface receptor signaling via STAT. In this network, taken from StringDB interactions, we annotated proteins here, each circle is a protein, based on proteins that are increased in protein abundance in pink or decreased in protein abundance in blue. However, many of the key proteins in JAK-STAT signaling do not change at the level of protein abundance. There's JAK1, as we just noted, but also JAK2 and JAK3, IL-10 and TYK2, IL-6ST, etc. If we augment our observation of changes in protein abundance with those that we observe changed at the proteoform level, we see a very different picture. ProMIS-OS detected 29 proteoforms that were modulated. Each of these highlights potential new biomarkers and also potential drug targets. For each of these, we can see which proteoform was annotated as increased in red or decreased in teal, as well as all of the post-translational modification sites that sit on this proteoform. In this picture, this combined representation of changes in protein abundance and protein state through proteoforms brings us again closer to what we know to functionally occur in inflammation in colon. Zooming out even one step beyond this, we can ask the same question not just for JAK-STAT signaling, not just for JAK1, but for a variety of critical drug targets in IBD, in ulcerative colitis. For this, we assembled an interactome, or the set of proteins known interacts with a list of different drug targets in IBD, in ulcerative colitis. From this set of interacting proteins, we asked 2 questions. What percentage of the proteins are modulated at the level of protein abundance? And what percentage of the proteins are modulated at the level of proteoform abundance? And in this graph here, what you're seeing is the percent increase in modulation at the level of proteoform versus protein. Or in other words, the added resolution that you can gain by looking at changes in protein state as well as protein abundance. JAK1, which we looked at in detail, is sitting here with around a 20% increase in proteoform coverage. However, many of our critical disease targets, such as TYK2, TREM1, IL-23, have even higher improvements in coverage by looking at the proteoform level. To summarize this section, proteoform modulation better aligns with known functional hubs than conventional protein abundance changes alone, highlighting how epiproteomics, again, brings us one step closer to understanding functional disease phenotypes. For our next use case, I want to look at SLE understanding, or systemic lupus erythematosus, in PBMCs, where epiproteomics provides a new lens on dysregulated pathways. Here, what I want to show is not only can we recapitulate known functional findings, but we can identify new mechanistic dysregulation that's less well known in the literature. I also want to note here that these data are from peripheral blood mononuclear cells, which is an excellent source of non-invasive biomarkers. It's cells taken from a blood sample, which enable us to identify biomarkers of disease or mechanistic insights without a tissue biopsy. Through a promissory analysis of these data, PTM resolution increased the number of differential features by 4-fold here. More critically, most of the changes we saw in protein modification are independent from changes in protein abundance. That means that we would not have known that a PTM was changed based on changes in protein abundance alone. There isn't more phosphorylation, for example, because there's more of that protein. Rather, they're decoupled, distinctly regulated processes. And we wanted to understand which mechanisms are being modulated at the level of protein abundance and which are being modulated at the level of post-translational modifications. And so to do this, we performed pathway enrichment on the proteins modulated at the level of abundance or the level of PTMs. Each of these circles represents a distinct pathway spanning the gamut of cellular processes, from cell communication in green, to immune system in red, to transport and metabolism in blue. This set of pathways here were detected by changes in protein abundance. This set of pathways here were detected both at the level of protein abundance and the level of post-translational modification modulation. However, the largest set of pathways here were detected as changing only at the level of PTMs. None of these pathways would have been identified as dysregulated if we looked at protein abundance alone. To zoom in on one example of this, I want to focus on two types of cytokine signaling. In the shared pathway, we have interferon alpha beta signaling, and in the PTM-specific pathways, we have IL-12 family signaling. Interferon alpha beta, or type 1 interferon, is one of the canonical, well-known hallmarks of SLE, and it's no surprise that we see this signal changing both at protein abundance and PTM state. However, IL-12 is a less well-known mechanism in SLE. And when we look at why IL-12 is enriched in these data, and it's enriched specifically at the level of PTMs and not protein abundance, we can look at all of the proteins that make up the IL-12 family signaling pathway. In these graphs here, all of the proteins in the pathway are listed on the y-axis. And here in black, we show the two proteins that are changed on the level of protein abundance, CA1 and STAT1. Two proteins out of a full pathway is not sufficient to drive statistical enrichment of this pathway in SLE. However, if we look at the lens of PTMs on these proteins, we can see a number of proteins have significant changes in a PTM in the protein, including JAK1, IL-6ST, and other proteins on this list. And through this list, we can clearly see why there's an enrichment of IL-12 family signaling. So by incorporating PTMs, we can identify novel dysregulated pathways, which serve both as biomarkers and novel mechanistic understanding into disease drivers. I now want to move to our last two use cases, focusing on urine as a source of non-invasive biomarkers. First, want to start by looking at patient journeys in lupus nephritis, which is a kidney disease that results as a complication of SLE, where we can capture post-translational disease regulation. In this cohort, we had two types of patients. We had patients whose kidney function improved when their SLE went into remission state, and these patients are shown in teal. And we had patients whose kidney function did not improve, despite the fact that their systemic SLE went into remission. If we look at the level of protein abundance in this PCA, we can see that we can't clearly distinguish between patients whose kidney function did not improve in gray and those whose kidney function improved in teal. However, if we, through unsupervised clustering of all of our post-translational modifications, look at patients, we can see that patients whose kidney function improved in teal are distinct from the PTM profiles of patients whose kidney function did not improve, suggesting that PTMs in urine can again better reflect the function of the kidney than protein abundance alone. And I want to discuss one specific feature that's driving this separation, which is the protein AMBP. On the level of protein abundance, while we have a slight increase in AMBP in non-improvers, it's not significantly so. And there are many non-improvers with AMBP levels equivalent to the improvers. Biologically speaking, while AMBP is encoded as one protein, it functions as two distinct halves. Following its translation, it's cleaved post-translationally into two halves, A1M, which protects from oxidative stress, and bicarin, which is an ECM protease inhibitor. And these two distinct halves function independently, but you can't detect them looking at the level of protein abundance as a whole. However, by looking at PTMs, we can distinctly monitor each half and what's happening to it independently. First of all, on AMBP, we detect a number of PTMs from many different types listed here. And we can see where each and every post-translational modification site sits across the amino acid positions on the protein, from the beginning of the protein till the end. Each circle represents a different PTM. Those that move to the right in this graph are significantly higher in improvers, and those that move to the left are significantly higher in non-improvers. And we've listed here the two halves of AMBP, the A1M half, which stops here, and the bicarin half, which starts here and goes to the end of the protein. And if you look at the PTMs sitting on the two halves, you'll notice very distinct patterns. A1M has a reduction in an age or advanced glycation end product, a stress-related glycation that occurs in many proteins in urine. By contrast, bicarin has two PTMs oxidation sites, which are increased in patients who improve. And this shows that the separation that we see, or the functional distinction of kidneys, is in part captured by our ability to distinguish between one protein with two distinct functions through the lens of PTMs. For the last use case, I want to discuss another urine cohort from chronic kidney disease, where I want to highlight how we can define more mechanistic biomarkers of disease progression. In this cohort, through reanalyzing the data, we had 4 distinct subgroups of patients. We had patients here in gray who have normal functioning kidneys, for all intents and purposes, healthy kidneys. And then 3 groups of patients with increasing disease severity, from patients with mild kidney disease in pink to severe kidney disease in red. And on the level of proteins first, we asked which features or which proteins are reduced with severe disease or increased with severe disease. And in this heat map, you can see in the top box, we have all of the proteins reduced in severe disease, going from red, which is high, to blue, which is low. And in these bottom 2 boxes, the opposite, proteins that are increased or markers of disease severity. But we can do the same thing on individual PTM sites and identify a wholly distinct set of features of PTMs, each of these serving as a potential marker of disease severity. Now, what's notable about the PTMs versus the proteins is when we focus in on CKD1 or mild disease severity. On the level of proteins, we see fairly homogeneous patterns. All of our patients in CKD1 state have fairly similar profiles of proteins. They are high in these features and low in these features. When we look at the level of PTMs, however, we see a more distinct gradient. Some of our patients look much more like healthy individuals, while some of our patients look more like severe disease kidney stages. And this suggests that PTMs may be a source of novel earlier biomarkers. These changes that we're detecting at early stages may help us to track patients' disease progress at earlier states. Beyond being earlier, I also want to show how these PTMs are more mechanistic. And to do this, I want to focus on one of the central processes that occurs during kidney disease, which is remodeling of the extracellular matrix in collagen, which ultimately disrupts the filtration barriers of the kidney. And when we look at many of the processes involved in ECM remodeling on the level of protein abundance, we see little to no enrichment. Each of these circles represents a distinct pathway in ECM remodeling, from extracellular matrix assembly here to collagen trimer formation here. If we zoom in on collagen trimer, each of these circles represents a protein in the collagen trimer pathway. The proteins in purple have changes in abundance in CKD, meaning protein is increased or decreased with disease severity. However, the purple proteins here are not sufficient, again, to drive enrichment of the collagen trimer pathway. protein abundance changes alone do not highlight collagen regulation in CKD. However, if we add on the lens of collagen oxidation, one of our specific PTMs from our broad profile, we have a very different picture. We've now labeled in teal all of the proteins who are changed not in abundance, but in oxidation state. And you can see many more proteins in collagen trimer formation are modulated either through oxidation or through protein abundance. And when we look back at our pathways, we see many more of the pathways in ECM modulation are significantly enriched in CKD, suggesting that adding in different levels of PTM regulation help us to uncover mechanistic changes that occur during the disease. Finally, this change that we observe is not specific just to chronic kidney disease. We observe the same thing in lupus nephritis. What we did here is we did a meta-analysis of all of our different cohorts of CKD and lupus nephritis. Here in these, for all of the proteins, each row is a protein, we see proteins that are increased in abundance or decreased in abundance. Notably, our collagen proteins do not change in abundance either in CKD or in lupus nephritis. However, if we again add on the lens of protein oxidation, we see the majority of the collagen proteins listed here have reduction in oxidation in urine in, say, CKD and lupus nephritis, suggesting oxidized collagen is a broad signature of ECM modulation in kidney diseases. Today, I showed you a number of use cases whereby Promise OS is a versatile solution to maximize insights. It supports multiple tissue types and biological fluids, does not require any specialized processing or enrichment. It's compatible with diverse mass spectrometry platforms and offers higher returns from every experiment by unlocking deeper insights, increasing site detection, and identifying novel potential biomarkers and targets. And we want to, going forward, leverage Promise OS on a broader scale. And to do that, we're assembling the Epitome, or a continuously growing database of clinically annotated samples from diverse tissue types and diseases. We currently, in our Epitome, have over 170,000 PTM sites from over 2,000 samples from 13 diseases across 10 tissue types. And these comprise both public data sets we've reanalyzed, as well as data sets we've collected ourselves from samples ourselves that we've run proteomics on. And the Epitome will serve as a future source of biomarker discovery, as well as mechanistic insight into different diseases. In general, Promise OS has broad use cases for many clinical questions. PTMs can aid in biomarker discovery, and by extension, patient selection through biomarker discovery, target discovery, indication selection and expansion, and dosing optimization. And part of the reason our use cases are so broad is because our tissue types that are applicable are very broad. We can use Promise OS on clinical samples from liquid biopsies, which are a great source of non-invasive biomarkers, such as PBMCs, plasma, or urine, as I showed today. We can also use Promise OS on solid tissues, such as colon or skin, brain, etc., to get better disease context into critical drivers of the disease. And finally, we can analyze the immunopeptidome, or peptides presented on HLA class 1 and 2, to find PTMs which may be a source of antigens in cancers. And with that, I want to encourage you all to collaborate with us to better drive impact in precision medicine. And I'm happy to take any questions. Thank you for your attention.
Thank you so much, Aaron. That was a really fascinating and informative talk. As with all the talks today, we've received a lot of questions from the audience, so I will move straight on to the Q&A now. What types of mass spectrometry data can be analyzed by Prospect? Are there limitations on instrument types or acquisition methods?
That's a really good question. So first and foremost, we can analyze both DDA and DIA capture methods, so data dependent and data independent. With data dependent, it's a very simple process. With data independent, we can, depending on the instrument type, either do a direct search with all of our PTMs or generate a DDA spectral library, which we can use to search against the DIA data. We're compatible with a wide variety of instrument types. And of course, the resolution of both the proteins and the PTMs is going to be dependent on the quality of the instrument used.
Wonderful, thank you. And another one, how can I use Promise OS for broad PTM profiling? What does a typical collaboration look like?
So we really like collaborations to start from a fundamental clinical or biological question, such as biomarker discovery for a given treatment or disease context. We would then identify the suitable cohorts to use, both publicly available data, but also data that we would collect together or data sets you would have on hand. And then we'd reanalyze first by augmenting, first by capturing what we have through Prospect, capturing all of the PTMs that are relevant for the disease context, and then using our epitome to augment and our reasoning to derive those actionable insights depending on the original clinical question.
Thank you. That's great. So another question, how might proteoform level insights such as JAK1 modulation in ulcerative colitis influence therapeutic targeting differently from protein level measurements?
So we're still a lot to learn on proteoform level, but it can influence it at least in two different ways. Way one is as a novel marker for companion diagnostics. The protein level alone, as we showed today, may not track with disease severity or response or disease state, but at the proteoform level, it would. And we can use this more precise marker as a diagnostic or response signature. Beyond that, in some cases, proteoforms may hint at mechanistic regulation. We may see specific PTM sites at an area known to turn a protein on or off or signal in some way. And this could give us mechanistic clues that we could eventually target to reverse disease processes.
Wonderful, thank you. And we have time for another one or two, actually. What functional insights can be learned from changes at the PTM or proteoform level?
That's really the Moneyball question. Because PTMs are so understudied, it's not as easy or as trivial as with protein abundance, where one can go to the literature and find 20, 30, 40 papers annotating where that protein is and what it's doing. So in part, that's why we're growing Epitome, to give us better functional annotation, clinical annotation of which PTMs are where and which processes they're associated with. Beyond that, however, we can use a lot of information at our disposal, such as structural information, by seeing where PTMs sit, or information we know about which enzymes are putting the PTM there, or which proteins bind at that interface. We can generate hypotheses for specific PTMs, how they might affect the protein, and ultimately what the functional consequence of the change in the PTM might be.
Thank you. And time for just one more. What are the most promising disease areas beyond immune-mediated conditions where epiproteomics could uncover novel biomarkers or drug targets in the next 3 to 5 years?
That's a good question. So, I mean, first of all, I'm biased, but I think PTMs matter for all biological contexts. But all joking aside, we focus today on autoimmune diseases and immune-mediated disorders. But of course, there's much information in the literature about the involvement of PTMs in cancer already, both through work we've done in the past and also through large studies such as CPTAC, where they've analyzed many different PTMs contributing to cancer. I think likewise, the next forefront is neurodegenerative disorders. As I showed way at the top of the talk, phosphorylated tau is emerging as a critical mechanistic driver and marker in Alzheimer's and other neurodegenerative disorders. And so I really think that we'll be seeing over the next few years, PTMs really emerging in that space as well.
Thank you. Thank you so much, Aaron. Unfortunately, that's all the time we have for questions today. Any questions that we didn't have time to get to will be sent to the speaker after the event. That just leaves me to say thank you once again, Aaron, and we'll be back soon with the next talk.